Hello and welcome back. Now we're going to move on to the central nervous system, the brain and the spinal cord. We just spent the entire last section talking about neurons and action potentials, depolarization, repolarization, and everything we need to know before talking about the nervous system. Now we're going to talk about the central nervous system, the brain, and the spinal cord. Before we do that, however, I want to talk about something that we talked about earlier in this course, was what are called neuroglial cells or glial cells. And those, if you remember, are support cells for the main cell of the central nervous system, which are neurons. Now, if you remember that neurons, if you think about it, are highly specialized in what they do. They're very active, they're very energy consuming, because what they're constantly doing is through their dendrites, taking up information, sensory information, which is being processed in the cell body. And we don't know if it's a conscious thing or not with them, but deciding, integrating, processing all this stimulatory and inhibitory information, excitatory and inhibitory information that's coming into the cell body, and essentially processing this to decide, is it going to propagate with an action potential or not? And they're so focused on doing this that they need support, they need extra help. Now think about most of the other cells in the body. Most other cells can do most of what they need to do by themselves. Neurons, on the other hand, are so highly specialized in what activity that they're doing and their functions, as they need extra support cells. So these neuroglial cells do this in the central nervous system. Now, on this graphic, there's 5 cell types. There are neurons. Here's the neuron in this light blue color, and this is a multipolar neuron, which we talked about before. And then we're going to see oligodendrocytes, astrocytes, microglial cells, and ependymal cells or ependymal cells. So let's start with oligodendrocytes. And again, the whole purpose of this is so if you're interested or you hear about a topic like astrocytes, which we'll talk about, that you can go into the literature and read up more about astrocytes, because you know where they are, you know where they live, you know what their function is, and you kind of have some sense of what it is that they do. But you want to learn more, you have that foundational knowledge. So oligodendrocytes, here's a couple nuclei for an oligodendrocyte. An oligodendrocyte in the central nervous system is like what a Schwann cell does to the peripheral nervous system, and that is they make and maintain myelin. So here you have the cell body of a multipolar neuron in the central nervous system. This would be its axon. And you can see that this extension, or sort of appendage coming out of the nucleus or the cell body of this oligodendrocyte, is wrapping itself around, kind of, I always picture it like a carpet, wrapping a carpet around this axon of this multipolar neuron. And that's myelinating this neuron. So this would be white matter within the central nervous system. Here, this little space is the node of Ranvier, which is a little, it's a space in between the myelination and allows for saltatory conduction, if you remember that as well. So it increases the speed of propagation of these multipolar neurons in the central nervous system by myelinating them so that it can go faster and skip this whole section of axon, if you will, and increase their speed of propagation. So those are oligodendrocytes, and you can see here in orange is the myelination surrounding the axons of these multipolar neurons in the central nervous system. Let's talk about the microglial cell really quickly. So in purple here, this microglial cell is the immune cell of the central nervous system, or one of them. At least it's been discovered. Microglial cells are phagocytic, and within their dendrites you can see they're very much, and we didn't talk about skin very much, but they're very much like some of these surveillance immune cells, in that they have these extensions to their dendrites so that they can sample a lot of area of extracellular fluid to see if there's anything foreign that might cause damage to the brain or the neurons. So microglial cells are phagocytic. They can phagocytose if they deem something that's foreign or damaging to the brain. They can phagocytose or ingest or engulf any kind of debris. So these are the immune cells, and they're sort of hypervigilant immune cells of the nervous system. And then up here is what I usually call them ependymal cells or ependymal cells. And we'll talk more about this next. Next we're going to talk about the meninges, we'll talk about cerebrospinal fluid, the choroid plexus, the ventricles, where cerebrospinal fluid is made, the functions of cerebrospinal fluid, and where it actually flows around the brain and the spinal cord. But for now, ependymal cells, or ependymal cells, are another of the support cells, the neuroglial cells of the central nervous system. What do? They make cerebrospinal fluid, which I've always been kind of fascinated in how they do this. It's fairly simple, what it seems, but they will take what's essentially certain molecules or substances from the blood. Blood is not cerebrospinal fluid, but some of the stuff in it is. And so they'll take the stuff that they need to make cerebrospinal fluid from the blood or interstitial fluid and turn that into cerebrospinal fluid, for example, certain ions or water. And so those are highly specialized cells, simple cuboidal cells, if you go back to when we're talking about epithelial cells. Simple cuboidal cells in their shape with certain appendages or extensions off of them in order to be able to make cerebrospinal fluid. And then lastly are the astrocytes, and I'm leaving astrocytes to last because they are multifunctional. They do a lot of different things. First of all, if you just look at this, I guess, hopefully semi-accurate representation of what these neuroglial cells might look like, is in this sort of tan color here. These astrocytes, you can see, are reaching and touching and synapsing with everything. That all of these axons here are synapsing with the cell body of the neuron. They are synapsing with, I'm sure we can find one here, down to the oligodendrocyte. They are synapsing with the ependymal or ependymal cells. So they are communicating with every single other cell in the central nervous system. Now why? What do they do? They are supposedly involved in regulating neurogenesis, the formation of new neurons. They are talk about the blood. One of the main things that they do is they help regulate the extracellular substance, the extracellular fluid surrounding all these different cells. Some of the things they can do is they can take up glutamate. When glutamate as a neurotransmitter is no longer utilized, they can take up glutamate and turn it into glutamine. They can take up ammonia, which can be highly toxic and highly damaging to the central nervous system. They can help to regulate and take up things like potassium and water to help maintain the homeostasis of the extracellular fluids in this central nervous system. Something that's very interesting is they can store and release glucose, and it's said supposedly in areas of the brain like the frontal lobe and the hippocampus, but for times where there's not enough glucose availability for certain neurons or certain parts of the brain, that astrocytes can store and release, fascinatingly, glucose during periods of hypoglycemia or excess glucose utilization, for example. It's also suggested, I've seen some papers talking about astrocytes may make and release small amounts of insulin by themselves, which is completely outside of the pancreas. Very interesting. Here's an example. If you want to look into astrocytes and in PubMed, look up things like hyperglycemia in astrocytes, and it's suggested that it alters their neurotransmission. Again, like I said, cultured astrocytes and insulin production, and how this is an area of research that's constantly growing, and by no means do I believe that we have even 1/10 of an idea of what all these cells, A, what cells are in here, and B, what some of these cells are actually doing. One of the things I find most fascinating about the astrocytes, however, is most people have heard about the blood-brain barrier. And the reason for a blood-blank barrier, like blood-thymus barrier, as you'll learn in level 2, or blood, like testis barrier, is when there's something within those cells that is really highly sensitive and can be altered significantly if the wrong things get into it, like the thymus, for example. So in the blood-brain barrier, it's the astrocytes that help provide what is this blood-brain barrier. Now in level 2B, when we talk about the cardiovascular system, there's 3 different types of capillaries. There's sinusoidal capillaries, which have sort of large gaps and holes in them. There are fenestrated capillaries that have smaller pores in them, let's just say. And then there's continuous capillaries, and those, as the name would suggest, don't really let much across. Now the reason why these types of capillaries, if you think back to when we talked about cellular junctions, we talked about tight junctions. And a tight junction is found on these continuous capillaries. And that means not a lot's going to get across. In the brain, we have continuous capillaries. But that's not enough to protect this very sensitive brain, or these neurons. So in addition to that, for the blood-brain barrier, these, they're not necessarily podocytes like you might see in the kidneys, which you'll also learn about. But you can see that these extensions of the astrocytes, they form sort of these layers connected to one another that are surrounding, this would be a capillary. So not only do we have a capillary here that has its own impermeability to it, but we also have these astrocytes that are surrounding this capillary, which then is forming what's called the blood-brain barrier. So one of the functions of the astrocytes is to be essentially the blood-brain barrier. So before jumping into the anatomy of the central nervous system, meninges, cerebrospinal fluid, the anatomy of the spinal cord, and parts of the brain. These are the neuroglial cells. Neuroglial cells are support cells for the central nervous system. There are 4 main ones that we know of: oligodendrocytes, astrocytes, microglial cells, and ependymal cells, or ependymal cells. The oligodendrocytes make and maintain myelin surrounding the axons of the largely multipolar neurons found in the central nervous system. The microglial cells are the phagocytic immune cell of the nervous system. The ependymal cells are the ones that make and release and secrete cerebrospinal fluid, which is incredibly important. And then lastly are these multifunctional astrocytes that regulate the extracellular fluid of the central nervous system, take up things like glutamine and neurotransmitters. They can take up ammonia. They can take up things like potassium and water to help maintain the right balance of the extracellular fluid, as well as providing glucose potentially in times of extra need of glucose for the nervous system, particularly or possibly in certain areas of the brain more so than others. And then lastly, well, I talked about neurogenesis, but they also form what is called the blood-brain barrier. So now let's take a look at the brain and the spinal cord.